Hello everyone. In this video we are going to see DNA replication in eukaryotes. If you go through my previous video, I have given a lecture on DNA replication in prokaryotes. Both the process are a bit similar, but the DNA replication in eukaryotes is bit complex, involves many enzymes, many enzyme complex and many factors which helps in assisting the proper replication of eukaryotic DNA. So let's get into the video. And it's just a little introduction. So replication is a process by which DNA is copied or it is duplicated to the newly formed cells, and for eukaryotes it takes place only once during the life cycle of the cell. Replication is highly efficient, occurring rapidly with great precision and at the appropriate phase of the cell cycle. So the basis of DNA replication in eukaryotes follows Chargaff's law, which states that adenine always pairs with thymine in double bond and guanine always pairs with cytosine with triple bond. And it is a very simple process. You see, when we go through a cell cycle, in G1 phase the replication will be initiated, and in S phase the DNA synthesis occurs and two daughter copies are produced with exact copies of DNA as present in the parental strand, and in G2 phase repair mechanism occurs. As similar to prokaryotic cell, the DNA strand is transferred to the newly formed daughter cells with greater integrity, and the semi-conservative mode of replication is also followed here. So as I stated before, it involves lot of enzymes, lot of replication factors and proteins to smooth the replication process of eukaryotic cells. DNA replication is divided into three phases when we saw in the prokaryote, but prior to the initiation step we have a pre-initiation complex step in eukaryotes. When you see pre-initiation complex, then which involves the formation of pre-initiation complex at origin of replication site. As we discussed before, as prokaryote, in eukaryote also the DNA replication starts in the origin of replication. It is also called as ORI site. There the pre-initiation complex will be formed, which is a step 1 in eukaryotic DNA replication. In step 2, the pre-initiation complex is converted into initiation complex via some other protein addition, and step 3 is elongation phase, in which the replication fork is formed and the DNA elongation nucleotide and the DNA elongation will take place, and last step is termination, which involves the telomere replication. So for the formation of pre-replicative complex, it is the first phase of eukaryotic replication and it begins in the ORI site, and these are the 4 enzymes which are involved in the pre-initiation complex formation. First one is ORC, which is the site where the enzymes recognizes and bind to the replication origin, and the second enzyme is cell division cycle C, which is also called as CDC6. It recruits other replication factors to the ORI binding site. And third one is CDT1, which is also called as chromatin licensing and DNA replication factor 1. It assists in loading the helicase complex into a DNA. As we discussed earlier, what does helicase enzyme will do? Helicase enzyme will helps in the unwinding of double stranded DNA into a single stranded one, right? That way this enzyme does that activity. And there is another complex called as mini chromosome maintenance complex, which functions as the helicase to unwind the DNA, and it enables a strand separation for replication process. And this is the chart which describes the steps involved in initiation. As we discussed before, all the enzyme complex will assemble in the ORI site of the DNA. Once they bind, they recruit the CDC6. Once CD6 binds with the ORI site in a ATP dependent manner, it recruits CDT1. CDT1 is required for licensing the chromatin for replication, followed by this CDT1 recruits CDC6. Finally, all the 3 proteins will recruit MCM. So, MCM will bind with the chromatin and all the steps in the initiation complex is occurring in the G1 phase of a cell cycle. And this is the diagrammatic representation of what I discussed before. All the factors will assemble in the ORC one by one, and it helps in the separation of a double strand into a single stranded one, which is also called as replication bubble. And you know, we have discussed many factors like MCM, CDC6, CDT1, right? There are two factors which is very important to control the replication process. As I discussed before, since cell cycle, in cell cycle the DNA replication takes place at only one time, it has to be controlled. So, first one is regulation of CDT1 activity. It is tightly controlled during the cell cycle to maintain the accuracy. It is controlled by a regulatory protein called as geminin, which inhibits the CDT1 during the S phase, thereby preventing the re-replication of the DNA. Once initiated, it has to be terminated. It should not revert back and do re-replication. This process is monitored by a geminin protein. In addition, with the inhibition of geminin protein, CDT1 is also regulated by ubiquitination and proteolytic degradation, which means once its role is finished, it has to be ubiquitinated, and subsequently it has to be proteolized, it has to be degraded, so that replication of DNA will be halted. And another one complex which need tight regulation is MCM complex. It's of 6 types, which is MC2, 3, 4, 5, 6 and 7, and this MCM complex is actually functions as a helicase which unwinds a double stranded DNA into a single stranded DNA, right? And it's a formation of helps in the formation of replication bubble, and it helps in the formation of replication for progression also. So, how it is tightly regulated is any one factors in this will be disrupted, so that the entire process will be stopped. So, these two proteins must be tightly controlled to control the re-replication of DNA. And the step 2 is formation of initiation complex. In step 2, the pre-initiation complex is converted into initiation complex with the help of these 4 enzymes, namely CDC45 GINS complex, cyclin dependent kinase, and DPTF4 dependent kinase. And the CDC kinase is responsible for activating the MCM helicase, which helps in the unwinding of DNA, and GINS complex helps along with the CD445 to facilitate this process for a longer time. And cyclin dependent kinase will initiate the phosphorylation process, which is necessary for activating the origins, replicating origins. Finally, DDK enzymes along with DDK complex is formed by the combination of CDC7 and DB4 phosphorylase MCM enzyme, and it activates the helicase and replication initiation. And as we discussed in the previous step, here also 2 enzymes which need to be strictly regulated, and their roles has to be very much notable. First one is CD45. It is responsible for the transition from pre-initiation complex to initiation complex. Any drugs which inhibiting the CDC45 will help in the prevention of DNA replication process, which is very important in the prevention of cancer progression. And next one is GINS complex. It plays an essential role in stabilizing the CD45 and MCM complex at origin of replication. If this GINS system is inhibited, it also helps in the halting of progression of DNA replication, which can also be used as a drug target. And actually the naming the GINS complex structure and meaning is, actually GINS complex is made up of 4 smaller subunits, which includes SLD5, PCF1, 2 and 3. These 4 complex proteins will join into the GINS complex, and it is responsible for transition of pre-initiation complex into a initiation complex 1. And step 3 is elongation. It is very similar to the prokaryote. In elongation process, what happens is all the complexes, all the enzyme complexes, protein complex which we discussed before will help to maintain the formation of replication fork, and DNA polymerase enzyme will help in the addition of newer nucleotide to the growing strand. On the other hand, ahead of this DNA replication, DNA nucleotide synthesis process, DNA supercoiling is also takes place, which may lead to breakage of DNA. As discussed in the prokaryote, there are two types of topoisomerase enzyme which creates a nick in the DNA growing DNA strand, so that supercoiling will be prevented. And replication fork is actually a Y-shaped structure, and it leads to a formation of leading strand and lagging strand. Leading strand is a continuous one which is synthesized in the 5' to 3' direction. So, there the leading strand will be a continuous one. Instead, the lagging strand will be synthesized in the 3' to 5' direction, and it requires multiple RNA primers and is a discontinuous strand. This is the diagrammatic representation of a leading strand and a lagging strand. Leading strand, as I said before, is a continuous one since it takes place in the 5' to 3' direction. DNA polymerase epsilon is responsible for addition of nucleotide to the growing strand, and primase enzyme is helpful in the removal of short RNA primer from the growing strand. Once synthesis is completed, primase enzyme is responsible for the addition of RNA primer to the growing strand. Primase will be helpful in the addition of primer to the growing strand, which will be followed by the growing nucleotides to be added in the growing strand. And elongation proceeds in the 5' to 3' direction. Once the leading strand is completely synthesized, the RNA primer which is initially placed in the growing strand is removed by RNase H enzyme. The gap will be sealed by DNA ligase enzyme. Lagging strand is a discontinuous strand. It contains multiple primer, and here the elongation is carried out by DNA polymerase delta. Once the leading strand is completely formed, there will be many gaps because the attached multiple RNA primers will be removed by primase enzyme, and the gaps will be sealed by DNA ligase. And the last step is a termination process. You know, in eukaryotic cell we have telomeres at the end. So, telomeres are synthesized by enzyme called as telomerase, which is which gets extended in the 3' direction of the parental strand. It has overhanging ends at the 3' end. This will recruit the telomerase enzyme. Telomerase enzyme is made up of a protein part as well as a RNA primer part. This RNA primer part will attach with the 3' overhanging ends in the telomere, and it starts synthesizing the remaining DNA molecules, and this protein subunit of telomerase is called as TERT, that is telomerase reverse transcriptase. It synthesizes DNA from the RNA template. In a previous step we discussed DNA will serve as a template for the replication process, but in telomerase enzyme alone RNA will be served as a template from which the newly grown DNA will be synthesized. Once the extension is complete, TERT detaches from the overhanging ends and your DNA synthesis will be completed, and this has been explained in this diagrammatic representation. As I stated before, in the 3' end there is the overhanging end which protrudes the telomerase binding, and it carries out the polymerization reaction, and it does step 3 is translocation. Once the full synthesis is over, the TERT complex will be removed and the whole DNA has been successfully synthesized, and here also fidelity of the DNA is maintained by DNA repairing enzymes. Thank you.